Today we're going to get into some fascinating scientific detective work, straight from the journal Neuron. And it's a wild story, really. It's about one of the absolute toughest cancers out there, a very clever disguise, and a potential new way to fight back, by using the tumor's own dirty tricks against it.
Okay, so first, every good detective story needs a villain, and our investigation starts with a type of brain cancer that has frankly been a nightmare for scientists for decades.
And we're not talking about just any tumor here. This is glioblastoma, or GBM for short. It's notorious. It is aggressive, it's invasive, and it is just incredibly difficult to treat.
In this quote, this really lays out the grim reality. Even when we throw everything we have at it, surgery, radiation, chemo, the cancer, it almost always comes back. On average, patients survive for only about 15 months. It's just devastating.
So that's the big question, right? What is its secret? What makes this cancer so incredibly resilient? Well, it turns out it might be getting a little help from a very unlikely source. So here's where the detective work really kicks in. The first major clue popped up when researchers started looking really closely at tumors that had already been treated but then grew back. They found something they really didn't expect.
You know, it was one of those patterns that just didn't quite add up at first. A specific type of healthy brain cell was showing up at the scene of the crime and in suspiciously large numbers.
And get this, when they identified exactly which cells were crowding around, they found they were oligodendrocytes. We'll just call them OLs. Now, these are normally the good guys, cells that help support our neurons. But study after study showed the same weird thing. The tumors that came back were packed with way more of these OLs than the original tumors ever were.
And it wasn't just how many there were, it was where they were. I mean, look at this. They weren't just scattered around randomly. No, they were clustered together, forming a perimeter right at the border between the tumor and the healthy brain tissue.
So this was the million-dollar question. What were they doing there? Were these healthy cells trying to form a barrier, trying to fight the cancer? Or was something way more sinister going on here?
To figure this out, the researchers had to work backwards. How did these AOLs even get there? Well, it turns out the tumor wasn't a passive bystander in all this. It was actively sending out a recruitment signal. It was calling for backup.
And they found the signal. The tumor cells were pumping out the specific chemical, a chemokine called CX3CL1. You can pretty much think of it like a beacon or a siren's call that's being broadcast out into the brain. And who has the perfect antenna to pick up that signal? The oligodendrocytes. They had this specific receiver on their surface, CX3CR1, that is tuned to that exact frequency. It's a classic lock and key system, and it draws them right to the source, the edge of the tumor.
So the tumor calls for help, and the help arrives. But this is where the story takes a really dark turn, because this isn't a rescue mission. It's a corruption.
Yeah, once these perfectly normal, healthy brain cells arrive at the tumor's doorstep, they enter this hostile environment. And that environment, it fundamentally changes them. Once they're lured in, the oligodendrocytes get totally transformed. They're not the same cells anymore. Scientists actually call this new state reactive OLs. Essentially, they've been brainwashed. They've been turned into unwitting accomplices for the tumor.
So what's their new job description? Well, it's pretty straightforward. First, they become this dedicated support crew. Second, they start pumping out their own chemical called CCL5. And here's the absolute kicker. This CCL5 acts like a high-octane superfuel. But it doesn't just feed the whole tumor. Oh no, it feeds only the most dangerous cells inside it.
And those cells are the glioma stem cells, or GSCs. Think of these guys as the queen bees of the tumor hive. They're a small, elite population of cancer cells that are super resistant to chemo and radiation, and they can regrow the entire tumor all by themselves. They are the reason this cancer always comes back. And this is where the whole conspiracy just snaps into focus. The corrupted OLs release the fuel, that's CCL5. The cancer stem cells have the special fuel cap needed to receive it, that's a receptor called CCR5. This secret supply line gives the most dangerous cells in the entire tumor exactly what they need to survive our best treatments and drive the cancer's deadly return.
Now, that sounds pretty bleak, I know. But uncovering a secret supply line like this is a massive breakthrough. Because once you know how the engine is being fueled, you might just know how to cut the fuel line. And that's the beautiful, simple idea for a new therapy here. Look, maybe we can't stop the tumor from sending out its call for help, but what if we can stop that help from actually helping? What if we can intercept that fuel delivery?
The strategy is it's brilliant. So here's the problem. CCR5 fuel docks onto the CCR5 receptor and powers up the cancer stem cells. The solution? Block that receptor. And the wildest part of all of this? A drug that does exactly that already exists. It's called Maraviroc. It literally just plugs up that CCR5 receptor so the fuel can't get in.
And the results from the lab? Well, they are incredibly promising. In studies with mice that had this type of brain cancer, treating them with Maraviroc, the drug that blocks this fuel line, led to a huge increase in how long they survived. I mean, you can see it right here in the data. It's a fantastic proof of concept. So let's just quickly recap the whole story here. Glioblastoma tumors actively recruit healthy brain cells to their side. They then corrupt them, turning them into a support crew that pumps out a chemical fuel called CCL5. That fuel is sent directly to the most dangerous cancer stem cells. And now we know that blocking this pathway with an existing drug shows incredible promise.
You know, this research really opens up a whole new way of thinking. For so long, we've focused on just attacking the cancer cells directly. But what if the key isn't just fighting the tumor, but also dismantling the corrupt ecosystem it builds to protect itself? This idea of sabotaging, it could be a real turning point, not just for brain cancer, but maybe for how we fight this disease, period.